In this class, I will discuss alternative complement pathway. In the beginning, I want to mention some proteins that are actually involved only in the alternative pathway. The example of these proteins that are only involved in this pathway are factor B, factor D and properdin. And in this pathway, C2 and C4 are not used, but C3 protein and C5 protein and the rest of the C6 to C9 proteins are actually used. In this case, this pathway starts without initiator protein. In case of classical complement pathway and the lectin pathway, I have mentioned there are initiator protein. Classical complement pathway, the initiator protein is C1q protein. And in case of lectin pathway, the MBL and phycolin are the initiator protein. But in this case, there is no initiator protein, but one thing is occur, that is called tick over. What is tick over? Tick over means in our plasma, C3 proteins are present in high concentration and they are produced constantly and degraded very rapidly. These are called spontaneously hydrolysis. Tick over means the C3 protein gets hydrolyzed spontaneously in the serum. The C3 protein present in the serum high concentration. So after it hydrolyzed, the C3 protein becomes C3 H2O, that is means C3 protein is hydrolyzed. Okay. Now this C3 proteins which is hydrolyzed in condition can bind with factor B protein. Now it attached with factor B protein. I am simply writing it as a B. When this factor B bind with the C3 that is hydrolyzed in condition, thus another protein called factor D can cleave the B segment into two fragments. In my previous classes, I have mentioned that the complement protein, when cleaved, they produce two fragments. One is A, that is called smaller fragment, and the B fragment, that is the larger fragment. In this case also, the factor D also cleave this B protein, and the B cleaved into two fragments called BB, that is attached with this complex, and BA released from the B fragment. This complex is called C3 convertase. In my previous classes, I have mentioned that the classical pathway and the lectin pathway merge into two enzymes called C3 convertase, C3 convertase and C5 convertase. In case of both the classical and MBL pathway, the C3 convertase is C4b and C2a, and the C5 convertase is C4b, C2a and C3b. But now you can see here is a difference between the classical complement pathway and lectin pathway with the alternative pathway. In case of alternative pathway, this C3 H2O Bb, this fragment is called C3 convertase. So now this is a difference. But this C3 convertase is also called fluid phase C3 convertase. Fluid phase. What does it mean? Fluid phase means this complex is in the serum. That is called fluid phase. And one thing I want to mention, in this condition, if there is no infection, this C3 convertase enzyme gets degraded very rapidly. If there is no infection, this C3 H2O Bb means C3 convertase protein gets degraded very rapidly. But if there is an infection, then what happens? This C3 convertase actually cleave the protein C3. As this name C3 convertase means it can cleave the C3 protein into two fragments, C3a and C3b. Okay. If there is any bacterial infection or bacteria present in the serum, this is a bacteria, then what happens? This C3b protein get attached with the bacteria, and now the factor B protein can also bind with C3b. As I have mentioned, the factor B protein can bind with C3H2O, means hydrolyzed C3 protein. the factor B protein also can bind with C3b that is attached with the bacteria. Now the same process occurs. This B complex, B complement proteins is degraded or cleaved by factor D and it becomes C3b Bb. This is a larger fragment so that it can attach with this molecule. Now this is another time it is called C3 convertase, but it is stable. This complex is stable, but here is a difference between this C3 convertase and this C3 convertase, because in this fluid phase the C3 convertase gets degraded very rapidly, but in this case a protein called properdin actually stabilize the C3 convertase enzyme. Stabilize. It means now this protein cannot be degraded very rapidly. So this properdin actually stabilize this C3 convertase enzyme. Now the C3 convertase enzyme cleave the C3 protein again. Now you can see here C3 protein cleaved first time. Now the C3 protein is cleaved for the second time. The C3 protein cleaved into C3a and C3b. Now this fragment C3b again attach with this fragment and form C3b. Now you can see this complex C3b Bb C3b. This is called the C3b Bb, and next C3b, this complex is called C5 convertase. Okay, and then the C5 convertase cleave the C5 protein into two fragments, C5a and C5b. Now the C5b starts to attach with C6, C7, C8 and then C9, that form multimeric C9 to produce a pole-like structure that called MAC, means membrane attack complex.